Hi everyone, today our team will be presenting our image analysis final project titled Plant Leaf Disease Detection using Image Classification. Our team members are Varun Boda, Ajay Kumar Reddy Rachala, Murlidhar Reddy, Sirisha and myself Tejeshwar Reddy Rentham. In this presentation, I'm going to walk about through the motivation behind the project, the dataset, the CNN architecture, we designed, how we built everything in MATLAB, and finally the result and insight we gain. Motivation and background. We started this project with a simple but important questions. Can deep learning help farmers detect diseases early just from leaf images? Plant leaf diseases are one of the biggest reasons for crop loss worldwide. Usually, diagnosis is doing manually. Someone looks at the leaf and try to identify the disease. There are three main issues with that approach. It depends heavily on the expert's availability. It is slow and sometimes subjective. Many rural areas do not have access to the plant pathologists, so early detection becomes a huge challenge. This is where image-based deep learning systems can help. A simple mobile app provided by CNN can scan a leaf and instantly tell whether the plant is healthy or injured. This idea is motivated our entire project. Problems defined and objectives. In simple words, the input to our system is an RGB image of a leaf. The output should be a disease label. For example, early blight, late blight, leaf mold, or healthy. We focused on three crops: pepper, potato, and tomato. Our main research questions was, can a simple CNN architecture achieve reliable accuracy on the PlantVillage dataset subset? To answer this, our objectives were build a complete pipeline in MATLAB, preprocessing and augmented thousands of leaf images, train ACSS from scratch, evaluate this accuracy and errors, analyze whether our approach can be used for real applications. Related work and literature. When we reviewed prior literature, we found two trends. First one is traditional approach, and second one is the modern approach. The first one, before deep learning became popular, researchers extracted hard-crafted features like texture, color, homographs, edges, and shapes. They, they were used machine learning classifier like SMVs, KNN, or random forests. And the modern approaches of almost all recent papers use CNNs or transform learning models such as VGG, ResNet, and or MobileNet on the full Planet Village dataset. Several studies report more than 95 accuracy, but one thing we found interesting was the most academic paper used very large internets, so we decided to check whether a small, lightweighted CNN trained from scratch could still perform well. That's become a important part of our study. Dataset. The PlantVillage dataset was perfect for this project. It contains over 54,000 labeled images, over 34 different crop diseases combinations. All images are clean, well centered, and have a resolution of 256 into 256. For our project, we selected pepper leaf diseases, potato leaf diseases, tomato leaf diseases. After combining of all of them, we ended up with 15 classes in total for training the and testing. Becoming the Because the dataset is already well organized, we were able to load it easily using MATLAB's image data store. It's a simple image. These are simple images from the datasets. You can see healthy leaf and the diseased leaf. You can also notice how diseased leaves look extremely very usually. For example, tomato early blight and late blight looks almost identical. Different two tomatoes diseases have overlapping texture. This helps us understanding only on why this classification problem is not as easy as it looks. Pre-processing and augmentation. To prepare the dataset, we applied several pre-processing steps. First, all images were resized to 128 into 128 into 3, which made training much faster. We also normalized pixel values to a 0 to 1 range to avoid overfitting and make the model more robust. We applied data augmentation, random rotation, horizontal flipping, slight variations in orientation. This limitation real world scenarios which leaves may be titled or captured from slightly different angles. MATLAB's augmented image data stores made it very easy to implement all these steps. The workflow from our system can be summarized in 6 steps. Load rows images from the PlantVillage folder, split them into training, validity, and testing set. Apply augmented and resizing. Fourth one is training the CNN using MATLAB deep learning toolbox. Evaluating the training model on unseen test data. And the final one is visualizing result using accuracy curves, loss curves, confusion matrix, and example prediction. The pipeline help us keep our process organized and systemized. See, now, this is the architecture we designed. It has 3 main evolution blocks. Block 1 is 32 filter 3 into 3 batch normalization, ReLU, max polling, and block 2 has 64 filters, same structure as block 1, and block 3 has 128 filters, and remaining has the same structure, but it is very deeper than block 2 and 1. After 3 convolution blocks, we added dropout with the value 0.5 to reduce overfitting, a fully connected layer with 256 nodes, and finally a softmax output layer for 15 classes. Our goal was to keep the model simply but still powerful through enough to learn meaningful patterns. Training configuration. To train the module, we've used the following steps, like MATLAB deep learning toolbox, optimizer from Adam, learning rate is 0.001, batch size is 32, the captures are 10, and validation frequency is every 50 iteration. We train everything on a single CPU, and even the training finishes in around 15 minutes. We want the project to be reproducible by everyone, and even without a GPU. Now, coming to the result, on the test side, our model achieved about 91.27% accuracy, which is quite impressive for a simple CNN training from scratch. When we observe the training curves increases slightly and cross 95% by the latest EPSA. Validating accuracy stabilized around 90%. Training and validity losses showed good convergence. The important point is that there was no major overfitting, which means our augmentation and dropfit layer worked well. The conclusion matrix help us. The confusion matrix help us understanding which classes were easy and which were challenging. Most classes achieved more than 90% accuracy individually, However, certain tomato diseases showed confusing tomato early blights versus tomato late blights, tomato leaf mold versus spectorial leaf blood spots. They took extremely similar even to human eyes, so the misclassification was accepted. Low constraint images or images with only one part of the images were contributed to the errors. Simple predictions. Here we can see the two examples from our prediction set. The first example, the model correctly predicted a paper bell bacteria spot leaf. The features on the leaf were clear and distinguishable. In the second images, a tomato leaf mold was misclassified as a pepper disease. This might be due to the leaf looking very similar in shape and texture to pepper leaf in some class. These examples help us understanding what improvements are needed. Future work. This project opens us several possible directions for future works. First, we want to try transforming transfer learning using model like MobileNet V2, ResNet 50 or EfficientNet. These models are known to perform extremely well on small data sets. Second, we want to expand the data set to include images expressed in real forms, environments with very lightning, background noise, shadows, and partial leaves. Third, we want to develop a mobile app prototype where farmers can take a picture and instantly get predicts. Finally, the technique like GradCam can help visualizing which part of the leaf within network is focusing on. This makes the system more expandable. Conclusion. To summarize the picture, we build a complete end-to-end image classification system in MATLAB. We trained a lightweight CNN on a 15-class subset of PlantVillage dataset. Our module achieved a strong 95% test accuracy. The system performed well even without deep architecture or GPUs. The result shows that deep learning can be a practical tool for early plant disease detection. Overall, this project helps us understanding not only CNNs and images processing techniques, but also how these methods can be applied in real-world agricultural problems. These are our main references we use it for a literature review and model design. They include papers on deep learning for plant leaf distribution and the original plant village dataset source demo. Yeah, the right side part is our code, and this is our approaches. You can see that approaches and time lapse approaches are running in the command window.
And as you can see, this is the code for entire project, and here is on the left side approach. So like we train images, which is around 28,893 images, and and here is the coding for the project, like from data preprocessing to loading images and splitting uh, data into training and testing. So usually we do as 80% 80% for training and 20% for testing. So here we define arguments plus resized data stores, and here is the simple CNN, and here is the training options.
Yeah, this is our code. As you can see, this is the example of prediction and matrices. After this, we're going to the graphs. Yeah, this is the percentage of our accuracy of our project, and this is a loss. This is a very minor loss to in our project. The accuracy is very good while we're stopping it. When we stop this, we get some images like these. If we get the same like the tomato leaf molds and prediction was tomato leaf molds, it's going to be the correct one. As you can see here, pepper bell bacteria is a true, and we got the predicted as the same, so this is a true one. As you can see, it is totally different from the true and the predicted one, so it is our loss. This is the only thing we lost. Yeah, thank you for listening it. We're happy to take any questions.